To uncover the enigmatic threads of Jewish genetic origins is to delve into a tapestry woven from ancient migrations, resilient survivals, and the shadows of history's upheavals. Imagine a people whose DNA holds echoes of a distant homeland, yet whispers of far-flung admixtures that challenge simple narratives. This is no mere tale of lineage. It is a cautionary saga of how empires, exiles, and isolations forged a genetic identity that defies easy categorization. For centuries, scholars pondered the roots of the Jewish people through texts and artifacts, but only in the modern era have geneticists unlocked the code hidden in our cells, revealing truths that both affirm and complicate the stories passed down through generations. What secrets lie buried in the double helix, and why do they matter today? Let us journey back to the cradle of this mystery, where science meets the sands of time, and uncover layers that historians and scientists have debated for decades. The story begins in the ancient Levant, a fertile crossroads of civilizations where the Hebrew Bible places the origins of the Israelites. Historical records, from the Assyrian conquest in the 8th century before the Common Era to the Babylonian exile in the 6th century before the Common Era, document a people rooted in the lands of Canaan. Archaeologists like Israel Finkelstein have reflected on how these early inhabitants blended with local Canaanite populations, forming a cultural and ethnic mosaic that predates the monotheistic traditions we associate with Judaism. Finkelstein's work, detailed in his book The Bible Unearthed, suggests that the Israelites emerged from within Canaanite society around the 12th century before the Common Era, rather than through a massive conquest as biblical narratives describe. This blending is not just cultural, genetic echoes persist. Yet, the Roman destruction of the Second Temple in the year 70 of the Common Era marked a pivotal diaspora, scattering Jewish communities across the Mediterranean and beyond. Roman historians such as Josephus Flavius chronicled this dispersal in his work, Jewish Antiquities, noting how survivors fled to regions like Italy, North Africa, and the Rhineland. These exiles carried with them not just scrolls and traditions, but a genetic heritage that would evolve through centuries of adaptation and adversity. What if these forced movements introduced subtle genetic shifts, blending Levantine roots with host populations in ways that still intrigue researchers today? Fast forward to the medieval era, and the puzzle deepens with the formation of distinct Jewish groups. In the Rhineland, small Jewish settlements emerged around the 9th century, invited by figures like Charlemagne for their mercantile skills, as recorded in Carolingian charters. But peace was fleeting. Pogroms, such as the one in Erfurt in the 14th century during the Black Death, decimated communities, forcing survivors into isolation. Historians like Elisheva Baumgarten, in her studies on medieval Jewish family life, have pondered how these events created genetic bottlenecks, drastic reductions in population size that amplified certain traits and vulnerabilities, much like a river narrowing and intensifying its flow. What if these traumas etched themselves into the very fabric of DNA, creating patterns of inheritance that speak to survival against overwhelming odds? Geneticists have turned to ancient remains to answer this, analyzing bones from medieval cemeteries in Erfurt, Germany, and Norwich, England. A groundbreaking study published in the journal Cell in the year 2022, led by researchers like Shamam Waldman and David Reich from Harvard University, examined 33 individuals from Erfurt dated to the mid-14th century. Their findings? These early Ashkenazi Jews displayed a mix of Middle Eastern and European ancestries, but with greater heterogeneity than seen today. One subgroup leaned toward Eastern European influences, possibly from Slavic regions. Another toward Middle Eastern roots, suggesting migrations from places like Bohemia or Moravia. This diversity hints at a founder event predating the 14th century, where a small group, perhaps just a few hundred, survived and expanded, blending ancestries in ways that echo the diaspora's trials. The study also identified 15 disease-linked alleles, mirroring modern Ashkenazi frequencies, underscoring how bottlenecks intensified genetic risks. But who were these founders, and where did their genes truly originate? The intrigue lies in the autosomal DNA, the bulk of our genetic material inherited from both parents, which tells a story of continuity amid change. Studies by Harry Oster and colleagues at Albert Einstein College of Medicine, drawing on genome-wide data from thousands of individuals and published in the Proceedings of the National Academy of Sciences, reveal that Ashkenazi Jews share a major component of Near Eastern ancestry, tracing back over 2,000 years to the Levant. This is complemented by 30 to 60% European admixture, primarily from Southern European populations like Italians or Greeks, likely from interactions during the Roman and Byzantine eras. A landmark analysis in the American Journal of Human Genetics in the year 2010 found that modern Ashkenazi genomes form a tight cluster, distinct from non-Jewish Europeans, yet overlapping with other Jewish groups like Sephardi and Mizrahi. Mizrahi Jews from regions like Iran and Iraq cluster closer to Levantine peoples such as Palestinians and Druze, with minimal European input, as shown in Bahar's comprehensive study in Nature in the year 2010. Sephardi Jews, descendants of those expelled from Spain in the late 15th century as decreed by the Alhambra decree of Ferdinand and Isabella, show a similar mosaic. Middle Eastern cores with Iberian admixtures, including subtle North African influences from Berber populations. What explains this pattern? Reflections from geneticist Michael Hammer at the University of Arizona suggest a model of patrilocal migration, Jewish men traveling as merchants or scholars and marrying local women in host societies, though endogamy, marrying within the community, preserved core lineages over generations. Hammer's team, in a study spanning multiple Jewish diaspora groups, estimated that admixture events occurred primarily between the 1st and 10th centuries, aligning with historical migrations along trade routes like the Silk Road. Delving deeper into paternal lines, Y-chromosome studies provide compelling evidence of ancient roots with intriguing admixtures. Hammer's research, involving over 1,000 men and published in Human Genetics in the year 2000, showed that more than 70% of Jewish males and about half of Arab males inherit Y chromosomes from shared paternal ancestors in the Near East, dating back a few thousand years to the Bronze Age. Dominant haplogroups like J and E, common in the Middle East and Southern Europe, dominate Jewish patrilines, accounting for 35 to 43% in various groups, as detailed in a meta-analysis by Nebel and colleagues. For Ashkenazi Jews, European contributions are modest, around 12 to 23%, often via haplogroups like R1a and R1b, which are prevalent in Eastern Europe and may stem from medieval interactions during migrations to Poland and Lithuania after expulsions from Western Europe. The famous Cohen modal haplotype, found in about half of self-identified Kohanim, priestly descendants, coalesces to a single male ancestor around 3,000 years ago, supporting biblical claims of patrilineal descent from Aaron, as explored in Thomas's study in the American Journal of Human Genetics. Yet, debates arise. A study by Sergio Tofanelli in the year 2009 questioned the precision of dating, noting that mutations can blur timelines by centuries. Still, the consensus, as articulated by Daron Bahar in Nature Communications in the year 2016, affirms a unified Near Eastern origin for most paternal lines, with admixtures reflecting diaspora interactions rather than wholesale conversions. For Sephardi Jews, paternal diversity includes higher frequencies of haplogroup G, linked to Anatolian and Caucasian sources, hinting at Byzantine-era movements. On the maternal side, mitochondrial DNA unveils another layer of intrigue, revealing stories of women who bridged worlds. Ashkenazi women trace over 40% of their lineages to just 4 founder haplogroups, predominantly from the Near East, such as K1, A1, B1a, HV0a, and N1b, as identified in Bahar's seminal work in the American Journal of Human Genetics in the year 2006. However, scholars like Martin Richards at the University of Huddersfield have argued for 65 to 81% European maternal ancestry in Ashkenazim, proposing that Middle Eastern men intermarried with European women during early migrations through Italy around the first millennium, as evidenced in a study in Nature Communications in the year 2013. This matrilineal assimilation model suggests conversions or unions in the Roman Empire, aligning with historical accounts of proselytism by figures like Flavius Josephus, who noted Jewish communities attracting converts. Countering this, Kevin Brooks' ethnographic work in his book, The Jews of Khazaria, emphasizes ancient Middle Eastern roots for many lineages, with European inputs like Slavic haplogroups V7a and H11b1 as minor additions from the medieval period. A recent study in Human Genetics in the year 2020 resolved some tension, finding less than 15% distinctly European maternal lines in Ashkenazim, supporting a predominantly Near Eastern founder population for both sexes, with admixture peaking around 1,200 years ago. For Mizrahi groups, maternal diversity is intermediate. Iraqi Jews descend from 5 women, mostly Levantine, while Yemenite Jews show 42% traceable to 5 founders, blending West Asian and East African elements like haplogroup L, possibly from ancient trade or the Himyarite kingdom's conversions in the 4th century. These patterns intrigue because they reflect founder effects, bottlenecks where small groups' genes dominate descendants, exacerbated by persecutions like the Black Death pogroms or the Crusades in the 11th century, which reduced populations and heightened genetic uniformity. One of the most debated secrets is the Khazar hypothesis, positing that Ashkenazi Jews descend from Turkic converts in the Caucasus around the 8th to 10th centuries, when the Khazar Khaganate adopted Judaism as a state religion, as described in Arabic sources like those of al-Masudi. Popularized by Arthur Koestler in his controversial book, The 13th Tribe, in the year 1976, it gained traction amid anti-Semitic narratives suggesting Ashkenazim are not truly Semitic. But genetic scrutiny has dismantled it decisively. Iran El-Haik's analysis in Genome Biology and Evolution in the year 2013 claimed support using proxies like Armenians for Khazars, but Bahar's team, in a rebuttal in the European Journal of Human Genetics in the year 2016, expanded datasets to include actual Caucasus populations like Georgians and Armenians, finding no detectable Khazar signal, less than 2% matching. Instead, Ashkenazi ancestry aligns with Middle Eastern, 50 to 60%, and European, 40 to 50% sources, with any Eastern influences predating the Khazars and likely from earlier Silk Road interactions or Byzantine Jews. Historians like Shaul Stampfer at the Hebrew University have reflected that lacking historical evidence for mass Khazar conversions, only elite adoption is documented. The theory serves more as myth than fact, often weaponized to undermine Jewish claims to the Levant. This revelation underscores how genetics can debunk folklore, revealing instead a story of continuity from the Levant, with admixtures as footnotes to endurance. Yet the dark aspect emerges in the genetic legacies of isolation and persecution, not as some hidden conspiracy, but as the scars of history. The medieval bottleneck, estimated to have begun around 1,000 years ago in the Rhineland and spreading eastward, intensified runs of homozygosity, long stretches of identical DNA from both parents, increasing risks for conditions like Tay-Sachs, Gaucher disease, or familial dysautonomia, which affect 1 in 3,600 Ashkenazi births for Tay-Sachs carriers. The Erfurt study found 15 founder alleles linked to these in 11th individuals, mirroring modern frequencies and dating mutations to around 700 years ago. This isn't a secret of malice, but of survival. Endogamy, enforced by societal exclusion and religious laws like those in the Shulchan Aruch from the 16th century, preserved identity amid expulsions from England in the late 13th century under Edward I, or from Spain in the late 15th century. Geneticist Noah Rosenberg at Stanford University, in his work on identity by descent segments published in PLOS Genetics, shows higher sharing among Jews than with non-Jews, a testament to resilience through isolation. Sub-Saharan African admixture, 3 to 5% in diverse groups like Sephardim, dates to 1,600 to 3,400 years ago, predating the diaspora and suggesting shared ancestries with Near Eastern neighbors like Phoenicians or through ancient slave trades, as explored in a study by Parfit and Igorova. Expanding the lens to other groups adds depth to the intrigue. Ethiopian Beta Israel, airlifted to Israel in Operations Moses and Solomon in the 1980s and 90s, show 80% East African ancestry via maternal lines like L0 and L3, yet Levantine ties through paternal haplogroups J and E, supporting oral traditions of descent from the tribe of Dan, as genetically confirmed in a study by Hammer in the year 2000. Indian Beni Israel and Cochin Jews blend indigenous maternal lines, 60 to 70% South Asian haplogroups M and R, with Middle Eastern paternal, J and Q, reflecting migrations around the 1st century, as per historical accounts in the writings of Josephus and genetic analyses by Bahar. These far-flung communities highlight Judaism's adaptability, but also the genetic costs of isolation, such as unique founder mutations in Indian Jews for conditions like congenital adrenal hyperplasia. As we piece together this genetic mosaic, questions linger. How much did conversions truly shape the pool? And were they more common than texts suggest? Reflections from anthropologist Nadia Abu El-Haj in her book The Genealogical Science highlight potential biases in interpreting data, warning against oversimplifying Jewish genes as a biological essence, when Jewishness is a social construct layered on biological history. No single marker defines it. Instead, principal component analyses show Jewish groups bridging Middle Eastern and host populations. Modern implications abound, from health screenings recommended by the American College of Medical Genetics, targeting over 20 conditions in Ashkenazim, to identity quests via consumer tests from companies like 23andMe, which have revealed unexpected Jewish ancestry in millions, sparking personal reckonings. In Israel, genetic data informs immigration under the law of return, but raises ethical debates, as pondered by ethicist Yael Hashiloni Dolev.